Hello everyone, this is Kamcev, and in this video I'll briefly highlight a paper that was recently published by our group in Science Advances, and the topic of this paper is on specific ion effects on ion transport in charged polymer membranes. Now I can't do this paper justice with a single video because it's quite a lengthy paper that goes into a lot of detail of transport in these types of polymers. And so I'll most likely have to split this up into several videos. And so this video is going to focus on the main takeaway message or the key point from the study. Ion exchange membranes are used in a variety of electrochemical applications because of their ability to selectively transport counterions or ions that have an opposite charge to the polymer backbone over coions or ions that are similarly charged to the polymer. This type of charge selectivity is critical for applications like electrodialysis for water purification. However, recently there's been an enormous amount of interest in developing ion exchange membranes that can discriminate between different counterions, so being able to separate ions that have the same charge. Currently, there are some monovalent selective membranes that are available on the market, and these membranes can separate, for example, monovalent cations from divalent cations. But currently there are no membranes that can separate between ions that have the same valence. And this type of selectivity is critical for applications such as critical minerals recovery, and this has motivated an enormous amount of research efforts towards developing these types of membranes. To make better ion selective membranes, we need an improved fundamental understanding of how the structure of the polymer impacts the transport properties of the membrane. For this, we always start with the fundamental mechanism for ion transport in these materials, which involves partitioning at the membrane solution interface and subsequent diffusion across the membrane down an electrochemical potential gradient. The overall selectivity of ion exchange membranes can therefore be decoupled into a partitioning selectivity and a diffusion selectivity. Partitioning selectivity is simply the ratio of the two ions in the membrane to that in the external solution, and the diffusion selectivity is the ratio of the diffusivities or the mobilities of the ions in the membrane. We have projects in my group on both of these phenomenon, but today I'll mainly focus on ion diffusion within the membrane and the diffusion selectivity. Ion diffusion in polymer membranes can be viewed as an activated rate process. In this framework, an ion has to overcome a standard free energy of activation to execute a diffusion step within the membrane. This process can be described by the Reimonn-Polanyi equation, which relates the diffusivity to the standard free energy of activation. This energy is further decoupled into an enthalpic component, which is the so-called activation energy of diffusion, and an entropic component. The other parameter that appears here is the diffusional jump length. And in our study, we're using the size of a water molecule for this parameter. So essentially, when an ion diffuses in the membrane, it displaces one water molecule in a diffusion step. There are some studies that use the membrane thickness for this parameter, but I don't think that's quite accurate because that would suggest that the ion is essentially crossing over the membrane in a single diffusion jump. Nevertheless, we can use the Reimonn-Polanyi equation to rewrite the diffusion selectivity term from the previous slide and show that large differences in activation entropies or activation energies can lead to high diffusion selectivities. The main challenge is we don't really understand how the polymer impacts these parameters, and so the main goal of this study was to shed some light on this phenomenon. For the experimental component of our study, we synthesized a cation and an anion exchange membrane using the commercially available monomers shown here. We really like working with these monomers because they allow us to synthesize relatively homogeneous membranes, which are ideal for fundamental structure-property relationships. Furthermore, we can very precisely control the charge and water content of these membranes, and so we made these values identical in the sodium and chloride forms of the membranes. These values are also very similar to values that you'd find in commercial ion exchange membranes, and so these are very relevant ranges. We then chose a suite of anions and cations for our study, as shown here, to really establish these specific ion effects in these membranes. We measured the diffusion coefficients of the 15 different ions in the membranes as a function of temperature to obtain the activation energy of diffusion. We then analyzed the activation energy difference, which is the difference between the membrane and the external solution. I believe this is very important to do because ions have intrinsic differences in their transport properties, and specifically the activation energies in solution. And so to truly capture the effect of the membrane, we should normalize by this factor. This is not often done in the literature, and I think this is a significant shortcoming in this field. The final results are shown here, where this activation energy difference is ordered from largest to smallest, and indeed we see quite a bit of a spread between the different ions. Some ions like cesium have very little differences in activation energies, and other ions like iodide have enormous differences. The spread clearly demonstrates that there are specific ion effects in these membranes. Now we can also look at the activation entropy of diffusion, and we found that there's a very strong correlation between the activation entropy and activation energy, and this is quite often observed when transport results are analyzed within this framework, and it's often called the compensation effect. What this means is that essentially if you understand the activation energy differences, then you should be able to essentially predict the activation entropy differences. Because of this, we're mainly focusing on the discussion of activation energy differences in this particular study. Whenever specific ion effects are reported in the literature, they're usually rationalized by either one of two phenomena, and sometimes both. The first one is ion pairing within the membrane. One could argue that ions that form ion pairs should exhibit higher activation energies for transport. The other one is ion dehydration in the membrane, where one can argue that ions that can more easily shed their hydrating waters should exhibit lower activation energies for transport. However, most studies that report on either ion pairing or ion dehydration often don't show direct evidence for these phenomena, and indeed it's actually quite difficult to study these phenomena within membranes. In our study, through our combined experimental and molecular dynamics analysis, we wanted to directly answer the question about ion pairing and ion dehydration. I don't have enough time to go into all the details in this particular video, and I'll actually make separate videos on these two topics, but the main conclusions are shown here. When it comes to ion pairing, we found evidence that small, charge-dense cations can form contact ion pairs with the fixed charge groups in the cation exchange membranes. And we found that there's no contact ion pairing in the anion exchange membranes, but it's actually solvent-mediated ion pairing. When it comes to dehydration, we found that most ions do not undergo dehydration within the membrane, and particularly anions in the anion exchange membranes tend to exhibit something we call hyperhydration. They're actually more hydrated than in solution. To determine if there's a particular ion property that can explain our specific ion effects in the membranes, we performed an array of Pearson correlation tests with 17 different ion properties. The results are shown in the plot on the left. In this plot, we can see that the ion softness exhibits the strongest correlation with the activation energy difference between the membrane and solution, which you can see in the plot on the right. Polarizability also correlates fairly well with the activation energy differences, and in fact, polarizability and softness are somewhat related, but different concepts. Notably, the enthalpy or free energy of hydration does not strongly correlate with the activation energy differences, and these properties have been used in the past to explain specific ion effects in membranes. The concept of softness can be used in the framework of HSAB, or hard-soft acid-base theory, to describe ion interactions. Hard ions are classified as small, charge-dense ions that have few but very strongly oriented water molecules, for example lithium and fluoride, and soft ions can be characterized as large, less charge-dense ions that have many but weakly oriented surrounding waters. And so according to this theory, when two hard ions have an opportunity to interact, they can do so by shedding some of their waters and forming contact ion pairs. When two soft ions have the opportunity to interact, they can do so through collaborative hydration, and they typically form solvent-shared ion pairs. If there's a mismatch and you have a hard ion with the soft ions, these typically will exhibit minimal pairing interactions because of their incompatible solvation shells. So in summary, in our study, we found that the cations in the cation selective membranes tend to undergo these hard interactions and sometimes form contact ion pairs, whereas the anions in the anion exchange membrane undergo soft interactions, which can lead to solvent-shared ion pairs and more restricted hydration within the membrane due to this collaborative hydration. To conclude this video, I'm going to share some final remarks about the study, and actually most of these are going to be in the form of questions. I think we learned quite a lot in the study about topics like ion hydration in membranes, ion pairing in membranes, ion-ion interactions that ultimately lead to specific ion effects in these types of charged membranes, but there's still quite a lot of unanswered questions. One question is, are these phenomena similar in membranes that have either low water content or very, very high charge content? The membranes that we use for the study are pretty typical desalination membranes. I'd classify them as sort of high water content, medium charge content membranes. Of course, as you dehydrate these membranes or add more charges, some of these effects can be altered or exacerbated. So these are things that we're working on currently in the lab. We want to know if we can use the fixed charge group softness to control specific ion effects within these charged membranes. We used a sulfonate anion and a trimethylammonium cation in this particular study, but we can change the chemistry of the ions that are attached to the polymer backbone. So based on our finding that softness is very important for these interactions, can we control the softness of the polymer to control these types of interactions? There's also ongoing study in this direction. We demonstrated that there's this compensation effect, this enthalpy-entropy compensation effect, where changes in enthalpy are also accompanied by changes in entropy, and they essentially offset one another. So any gains that you might have for diffusivity in terms of enthalpy, they're offset by reductions in entropy. So how can we overcome, or how can we break this enthalpy-entropy compensation effect to achieve extremely high selectivities is very much an open question. And then finally, do these results extend to equilibrium ion partitioning and the related selectivity there? We focused on ion diffusion, but as I mentioned earlier, the entire transport process includes ion partitioning at the interface. Are these effects observed with partitioning? And also, are there trade-offs between partitioning selectivities and diffusion selectivities? So these are questions that are currently occupying our lab, and I hope to share more in the coming years.